Hi everyone. Let's just wait for maybe 2-3 minutes. A lot of people haven't joined yet. Okay, we'll get started. I'll just share my screen. Let me know if you can see it. I hope you can see my screen. I think you guys see it. Okay. Okay, then I'll get started for today. So, in your week 4 of coursework, you guys studied about mitosis in greater detail, and also the various different stages of the cell cycle, how they're regulated. You studied about CDKs and cyclins, maturation promoting factor, anaphase promoting complex, and how the CDKs and cyclins sort of go up and down in their level during the cell cycle, and that allows the progression of various phases of the cell cycle, one after another. And I think you also had guest lectures covering cytokinesis and other topics. So that must have been very interesting. So what I thought I'd start with is a recap of what we had actually seen, I think the last to last lecture, where I had gone through some fluorescence images of cells that were undergoing um, mitosis. And once we've recapped that, there's a few topics I thought we could take a glance at together and look at a few questions. Um, yeah, while we're doing that. Yeah. So if anyone has any trouble hearing my voice or seeing the screen, then just let me know. And obviously, when I read out the questions, as you know, it's great if you could unmute yourself and say what the answer is. It's okay if you're wrong. I'll obviously explain the premise entirely. Yeah. Okay. So here you can see these are HeLa cells that are, so the green staining, that's the microtubules. You have read now a lot about how important microtubules are in the way they organize themselves while the cell is undergoing mitosis. And they're important in not just helping the cell deliver different materials across and deliver vesicular cargo from one region to another, but of course forming the spindle apparatus that helps chromatids separate from one another, right? So here you can see in this video how in this cell here they're separating out and becoming two separate cells. Similarly here as well, play it again. They're forming the metaphase plate and now they are being pulled apart. And you can see the dissolving spindle fibers in between as well. And the rest of the cells that are not dividing, they are much flatter and they're not as rounded in shape. And that's an indicator that right now they are in interphase. Obviously, that's also very clear because their chromosomes are not condensed. Their nuclear material is much more diffuse looking than in this dividing cell, right? So this is routinely taking place in cell cultures, and this is a great way to visualize it. I had also shown you earlier on these images of your microtubules, also in green, and your chromosomes and your sister chromatids here. They are condensed and in blue. And then you have the pink kinetochores, right? So the kinetochores also is something that you studied in the course recently, right? Where you have these pink, sorry, these kinetochores that help the spindle apparatus attach to the centrosome of the chromosomes, and they help with aligning them at the metaphase plate and eventually pulling them apart. So this is another great fluorescence image of that. This is mitosis taking place in rat kangaroo epithelial kidney cells from this really great resource that I have linked at the bottom here. So I'll go through these phases individually um, right now. So this is an image of an cell in interphase. So you have your nuclear membrane, you have your chromatin that's not condensed yet, a centrosome hanging out near the nucleus over here, it's aster, and you have your cytoplasmic microtubule network that's generally sort of giving structure to the cell and helping with transport, but it is not polarized in a way that you see during mitosis. So obviously during interphase, we know that the cell, uh, whether it's G1 or G2 or S phase, essentially it's in the process of preparing for mitosis. So either it is um, preparing various components, preparing to double it, um, or it's increasing in size like in G2, or it's actively undergoing DNA replication and DNA synthesis so that you're duplicating the number of chromosomes so that they can separate out evenly. Next is prophase, of course. So that's your first phase of your entire M phase or mitosis phase. And here you have the nuclear material that's starting to condense and you can see it a lot clearer. And you have this early mitotic spindle forming here. The centrosome is polarizing in this one. And the nuclear membrane is going to start breaking down. It hasn't broken down completely yet, so because you can see that the chromosomes are not completely free to sort of roam around in the cytoplasm. And then in pro-metaphase, essentially you have complete nuclear breakdown. So you can see that now these guys have escaped. These condensed chromosomes are free to interact with the spindle fibers from coming from the different spindle pores. You have your astral microtubules and the ones that are extending across, the ones that are anti-parallel in between. right? And then these condensed chromosomes and your kinetochores are where they're actually attaching. Um, and then you have very obviously you have your metaphase here where your chromosomes have aligned at the middle, and you have these very clear spindle fibers from the spindle pole. And the cell is making sure not just that all the chromosomes have aligned here, but also that each chromosome is correctly um, attached to spindle fibers such that you don't get some ploidy issues when the cell divides and you have unequal segregation. So there are many checkpoints here as well for this. And now you have anaphase. So this is an early anaphase or anaphase E here, and this is where you have the shortening of these. So you saw how, in your coursework, you saw that there's a depolymerization that starts taking place and that shortens these and starts creating a tension here that eventually pulls the sister chromatids apart. And of course, there are many biochemical signals here as well, right? Like the cleaving of securin and separase enzyme coming out and managing to cleave at the centromere such that the chromatids can separate. So I'll go into all of that in more detail later on. So these things, these biochemical events, along with several mechanical events, are allowing these chromosomes or the daughter chromosomes to separate out into two new cells. And in anaphase B, essentially, it's just further separation, further lengthening, and getting much closer to telophase. And once we are beyond anaphase, this is a picture of late telophase. So a lot of the cleaving here in the middle has already taken place, and you have two separate cells. And now the cells will obviously have these signals to start going back into G1. So you want to start rapidly decondensing your chromosomes as well. You'll see a nucleolus start to reappear, and eventually these cells will completely separate out. And yeah, in the process, this is when they separate out in the process of cytokinesis, and you have two completely separate cells now. So these are some really great images, and they go into more detail on this website as well, so you can check it out in your time. So moving on from here, I hope no one has any questions. If you do, please let me know. I can explain anything. Um, so I thought I would talk a little bit about the cyclins and CDKs that regulate the cell cycle this entire time. So this is all from Khan Academy. This is a really nice article that they have that really simplifies it all and helps you sort of conceptually visualize it. So you can check it out at this link. So the levels of the different cyclins that we know, you can see how they are varying a lot across the cell cycle, right? And a typical cyclin, it's present at low levels for most of the cyclin, and when it is needed, like for instance here for the G1 to S phase transition, you need you need a cyclin at that point. So at that point, the G1S cyclin is going to increase, and you're going to have a peak. Similarly, the M cyclin to enter into the mitotic phase, you need a higher amount of M cyclin, so that level is going to increase. Interestingly enough, G1 cyclins are unusual in that way because they seem to be needed in one way or another for pretty much the entire cell cycle. So their distribution is much different from the other ones. Yeah. So, okay. So now we know that cyclins, they bind to CDKs, right? And that is what drives the cell cycle forward. So now a CDK on its own, it's a kinase enzyme. So it's something that adds phosphates to different substrates. So on its own, the CDK is actually inactive. But when a cyclin binds to it, it can activate it and make it functional. So this happens in two ways. Binding of the cyclin can activate the kinase activity of the CDK. So now it can go ahead and use um, ATP to add phosphates to its substrates. And the second thing it also does, it helps in targeting. So it targets the CDK or the CDK cycline complex to the correct proteins for that phase of the cell cycle. So an example of this would be the G1S cyclins, for instance. They, when they bind to some CDKs associated to S phase targets, they activate the CDK and also um, essentially localize it or sort of point it towards the S phase targets, which are proteins that help with promoting DNA replication. So S phase means that's what we want to do. We want to replicate DNA, replicate organelles, prepare more stuff so that the cell can double everything it has. So you have these targets getting phosphorylated by an activated CDK. Similarly, M cyclins send CDKs to M phase targets. For instance, for nuclear membrane breakdown, the breakdown of the uh, yeah, all of the nuclear membrane components. Yeah. So that's how this is working, and it's a very complex process. Obviously, there's not just one CDK. In yeast, there's just one, but in say mammals and stuff, we have many more of them, and they are not redundant. They have various very spatiotemporally regulated steps and functions. So one famous example that you guys heard about in your coursework is this MPF, right? Which is the maturation promoting factor. It's something that, was investigated in, the 1970s, and then finally in 1980 they realized, okay, so MPF is essentially an M cyclin bound to a CDK. So it's this complex that is then useful for phosphorylating various targets that eventually lets it go into M phase. Okay. So M-cyclin binds with CDK that allows it to phosphorylate its various targets because now the CDK is activated, and then the M phase can begin, your spindle can form, chromosomes can condense, and the nuclear membrane can break down. So this is one of the ways that the MPF is involved here. And of course, aside from MPF also, you have other important complexes that are involved in the progress of the cell cycle. So one important one is this anaphase promoting complex or cyclosome, so APCC. And it's a protein complex that causes M cyclins actually to start getting destroyed. So M cyclins are so this MPF is actually triggering its own destruction by activating APC, which will lead to the destruction of M cyclins, and that allows it to go into anaphase. Because if you have a lot of M cyclins, as you saw, M cyclins are higher in amount um, at the start of mitosis. It pushes it into M phase. But once you want to come out of it, once the cell is nearing to the end, you want to make sure that the M cyclin level goes down. So MPF is part of that cycle itself, and it can decrease the decrease the overall level of M cyclins to a point where then you only have the G1 cyclins in the major amount. So this is what this destruction of M cyclins would push the cells out of mitosis and allow the new daughter cells to then enter G1 phase, where obviously if there's M cyclins, you won't be able to go into that phase. And of course, they're also very important for making sure that they destroy these proteins that are holding the sister chromatids together, because how can anaphase progress if the chromatids are still stuck together? So we need to be able to cut up that cohesin complex that holds the chromatids together and allow them to separate out. Now, APCC, you might think, okay, this is one of those kinases again. So it's not actually, it does not add phosphate groups. It actually adds a ubiquitin. So you've, studied about the ubiquitin proteasomal system now, right? So you're quite familiar. So this is an E3 ubiquitin ligase. It adds a ubiquitin moiety onto a protein and targets it essentially for degradation, right? So one example is how it allows the complex holding this the two sister chromatids together, how APC is involved in that, and its ubiquitinating function is involved in that. So in the next slide, I think, yeah, the APC, it uses ubiquitin to tag and trigger the separation of the sister chromatids. So if it's getting the right signals, it's been activated correctly, it actually is ready, it can actually destroy cohesin and allow separation of the chromatids. So how does it do it? So there is this protein called separase. It's an enzyme, okay? And typically on its own it is inactive because securin is binding to it. So what does APC do once it's been activated? It adds a ubiquitin moiety to the securin, and that targets it for degradation. So now securin is out of the picture. So separase is active and it can go and chop up cohesin, which is one of its substrates. And once cohesin is chopped up, there's no glue holding these guys together anymore. So the chromatids can separate. So this is one very important function such that anaphase can be, and the cell can go into anaphase. So this is a video. This was actually part of your course. I think it was towards the end of one of your course videos, but I thought that with the commentary, it is very useful to see. It's from Drew Berry, and I will include the link when I post the video and the slides.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
Right. So I hope that was interesting, and uh, reflects it really reflects the way that the entire cell, including all of its force generating mechanical, chemical, and biochemical elements, comes together to make sure that the chromatids can separate evenly, right? Because none of these microtubule related events would take place if the cyclins and CDKs were not binding and cycling in their levels throughout the different phases of the cell cycle. So, yeah. So, I think we can move on to a few questions now, and you guys can call out the answer. So, an easy question to start off: during which phase of mitosis do the sister chromatids separate and move towards the opposite poles? You guys know? I hope you can hear me.
Anaphase.
Okay, that's right. That's great. Uh, it is anaphase, you're right. So, very good. Um, yeah. All right, here's another question. So, which of the following proteins is primarily responsible for depolymerization of microtubules during anaphase? Anyone know? If you want, you guys can answer in the chat as well.
22 तारीख है ना.
Okay. Uh, all right, so maybe I'll just tell you guys the answer. So it's actually kinesin 13. So there are many kinesins. This one is not there in yeast actually. This is there in, I think, like mammals and stuff. But anyway, there are analogs of this. So kinesin essentially is the answer. So particularly in this question, kinesin 13, it's a protein that's very important in directly regulating microtubule dynamics, and it's a protein that induces depolymerization, and very uniquely it does this through both ends of the microtubule. So that's not always the case with all kinesin family members, and this activity actually allows it to coincide with their cellular localization and with their ability to regulate microtubule dynamics and control the spindle assembly process and also the kinetochore to microtubule attachment process. So kinesins in that way are very important for that. And in general, the kinesin superfamily, as you know, and there's a video on this later on in the slides as well. These are molecular motors, right? So what do? They couple ATP hydrolysis with force production. So it was originally thought that all the kinesins, they actually just translocate unidirectionally in just one direction along this lattice of microtubules. But actually, with more extensive studies, people observed that this kinesin 13 family members actually depolymerize microtubules from both ends. And this was a, quite a challenge to the idea that people had initially. And then since then, it's become more clear about various kinesin family members and how they regulate microtubule dynamics. But most of them appear to do this only from one end. So in this aspect, kinesin 13 proteins, I thought it would be interesting for you to know. They are how do they do it? So they essentially um, bind to one end of the microtubule and they induce a conformational change at that end. And this leads to, as you guys had seen, how it's microtubule catastrophe, right? And then the kinesin 13s, with the help of ATP, can cause like they can diffuse towards the end and then they can consistently start depolymerizing from that end, but they manage to do this from both ends because of the way because of the way that they are organized, because of the way their domains are organized. Okay, I think something happened. Okay, all right. No, sorry. Yeah, so they essentially just bind to both the ends by changing the
मैम
um,
फास्ट आंसर इज anaphase.
sorry.
Fast answer is anaphase.
Yes, yes. First answer is anaphase. That's correct. Yeah. And the second one, which we're discussing right now, is kinesin 13. Right? I was just talking about how kinesin is important for depolymerizing the microtubules from both ends, and that's very important during anaphase, along with the activity of other kinesins and the activity of dynein as well, which helps with moving various cargoes to make sure that we can generate the right amount of force and tension that allows the chromatids to separate. I think I have a little bit of an internet issue today, so just let me know if you can't hear me in between, yeah? Okay, so the next question is, the mitotic spindle is primarily composed of which of these things?
Ma'am, microtubules.
That sounds right. Does anyone else have any other answer? Or does everyone agree with microtubules? Seems everyone is in agreement. So you're absolutely right. It's composed of microtubules, like we just saw. So there are, as you had seen, I think it was mentioned in your coursework, various different ways in which the spindle apparatus is oriented. So you have those astral microtubules, right? When you visualize it. And then you have the ones that extend across the cell and bind actually to the kinetochore. And then the ones that cross one another antiparallelly at the center as well. Okay, next question. So in animal cells, the contractile ring that's responsible for cytokinesis, what is it made of?
Option B.
Does anyone have any other answers? I think everyone agrees. So it's correct. Very good. It is actin and myosin. So actin, as you guys know, there are various important components of the cell cytoskeleton. You have your actin cytoskeleton, you have your microtubules, and you have your intermediate filaments. So actin is a very important cytoskeletal component of different cells, and it performs a whole myriad of functions, right? It's important for the shape, it's important for migration or moving of the cells, it's important obviously for this contractile ring that allows the cell to undergo cytokinesis. And myosin is another, just like how you have kinesin and dynein that are part of the molecular motor mechanism that travel on these microtubule filaments. Similarly, here you have myosin, which is also another force generating molecular motor that works with actin filaments or filamentous actin, F actin, and can generate force and waves of actin in different cells. So in this case, this contractile ring that is important for, sort of closing in on that boundary and making sure that the cells separate during cytokinesis, that is with the help of force generated, contractile force generated by actin and myosin. So great. So looks like you guys have the concepts in one here. I think I have some more questions. Right. So, which of these checkpoints ensures that the chromosomes are properly attached to the spindle before proceeding to anaphase? This is very obvious.
Option A.
Yeah, you're absolutely right. I should have put some less obvious options here, but that's very good. It is the spindle assembly checkpoint, and that's very important in making sure that you don't have ploidy issues after you have separated out the chromosomes. Which enzyme cleaves cohesin to allow cystochromatid separation? So I just spoke about this.
Phosphates.
Try again.
Option A.
That's correct. So I mentioned earlier on when I was just going through this concept a little bit in short, that separase is an enzyme that is inactive when it's just hanging out in the cell on its own. And there's this other protein securin that's bound to it, and that's what is making it inactive. then what happens is that when this anaphase promoting complex, APC, or the cyclosome is activated, it goes and adds a ubiquitin moiety to securin. So securin is gone now. So separase is active, and that is what goes and cleaves cohesin and allows the sister chromatids to separate. So I hope that's clear now. So this enzyme here, the answer is separase. Okay. All right. So the next one, the nuclear envelope breaks down during which phase?
Chromatophage.
That is correct. Great. So you're right. It is during prometaphase. That's when it completely breaks down. But of course, during prophase, the process starts and your lamins and all start breaking down a little bit. But by the time prometaphase is done, all your lamins have gone into complete dimer form and your nuclear material is free. Oh, I have more questions. Okay, so these are some fill in the blanks. So the enzyme dash phosphorylates various proteins to drive cell into mitosis. This is generally sort of a question.
NCDK.
Yeah, that's correct. Yeah, your M cyclin and CDK complex here in this case would be the correct answer. So that's great. All right. This protein complex, it holds the sister chromatids together until anaphase. So we were just talking about it. Anyone know? We were just discussing about some proteins and complex that helps hold the sister chromatids together. Yep,
Cohesion.
that's correct.
Cohesion.
And the person who messaged as well, that's completely right. It's the cohesin protein complex. So that's great. Good that you guys remember that now. Okay, next question. The mitotic spindle is organized by the dash in animal cells.
Nuclear organizing complex.
You're close to the right answer, but the name is little bit wrong. Think really hard about what the name was.
Centrosome.
Yes, good. So centrosome, and the person who said nuclear organizing complex, I think you meant microtubule organizing complex or MTOC, right? So the centrosome acts as the mTOC in the cells, right? And organizes the microtubules into the spindle fibers, right? So centrosome is completely right, and the function it's carrying out, because the centrosome has other functions, the centrosome is carrying out the function of an mTOC in this case, right? So great. Next question is the chromosomes align at the center of the cell during which phase? Easy one.
Metaphage.
Okay, great. So I think that one doesn't need too much explaining. All of you remember from your coursework as well as my couple of first slides that you have your metaphase plate or your equatorial plate forming during metaphase where all the chromosomes align. Okay, next question. In animal cells, cytokinesis occurs through the formation of a dash by actin filaments.
Contractile ring.
Could you say it again? I couldn't hear your answer. I think someone said it, but I wasn't sure. I couldn't really hear. But does anyone else know?
Contractile ring.
Yeah, I guess that could be an answer. So contractile ring is the force generating complex. So that is what drives the formation of this, what is the answer, which is the cleavage furrow. So the furrow is formed, right? That is driven by the force from the contractile ring. So I guess that's another answer too. Good job. Cool. So I think another one or two questions are there here. So the region of the chromosome where the spindle fibers attach to the chromosome, what is that called? Do you guys know? Try to remember this image I showed at the start of the presentation, right? There were these blue DNA ka image, and then there was the green microtubules, and then you had a pink dot-like structure on top of the centrosome. So it is some specific, very highly protein enriched structure. Right. Oh, okay. So people are messaging also. Hmm. Yeah, someone said centrosome. So this is actually attached to the centrosome. The next person is right. It's the kinetochore. Very good. So yes, the answer here is the kinetochore. Okay, that is where the spindle fibers attach to the chromosome at the region of the centrosome. So great. Good job. And the last question here. The return of chromatin to its less condensed state starts occurring during which phase? Anyone know? You can message or unmute and say, up to you.
Telophase.
That is correct. Very good. So obviously, the cell needs to go back to interphase. So once it's done separating out, the daughter cells will go into interphase until they need to divide again. So by the start of telophase, there are signals to start decondensing the chromosomes during telophase. Yeah. So good. So I think the basics of everything related to mitosis here is covered up nicely. So we can move to the next topic that I thought I'd just go into a little bit of detail in, because it's interesting that there could be a protein that is so highly pleiotropic. So in this case, this is the PLKs. So they were mentioned, right, in your coursework as well, that there are other kinases aside from your typical, cyclin CDK complexes and all that. PLKs, Aurora A, Aurora B, right? So PLK, I just chose one of them. This is a very important family, and you can see on the left from this image I took from this paper that they have so many functions, and there are different members of the PLK family as well, PLK 1, 2, 3, 4, and they, based on the color, you can see how they have different functions that are important for uh, the cell cycle. So PLKs are polo-like kinases, and they're called polo-like because they have this domain that comes from this polo box. Essentially, it was something that was discovered earlier in, I think, Drosophila earlier. And so they are a family of serine threonine kinases that have a very important role in the cell cycle progression and centrosome cycle also, because we know that the centrosomes also double, just like how our DNA doubles, centrosome also requires doubling, where you have your uh, yeah. So you have the mitosis and cellular, and it's also important in cellular response to DNA damage. And so initially it was discovered via a bunch of genetic screens, and a lot of this work was done in yeast initially, and it was discovered as CDC5 and Polo. CDC5 in budding yeast and Polo, and yeah, I was right, in Drosophila. And PLK1 is found in several locations throughout most of the cell cycle. So actually, just like how your cyclins and CDKs cycle through the cell cycle and their level as well as localization changes, here also PLK1 is restricted to specific stages and locations, whereas PLK1 is a little more dynamic that way. It's present the entire time, and also it's kind of like the G1 cyclin, right? In that way. So the main PLK1 functions here is it starts in G2, during which it localizes to the centrosomes. So you find G2 here. It's important here for making sure that there's mitotic entry is taking place properly and CDK activation is happening. It's important where it's found near the centrosome. It's important for centrosome maturation and checkpoint recovery. And then later it's also found during pro-metaphase and metaphase. So you can see that it's important for microtubule kinetochore attachment as well. It's this kinase. It must be phosphorylating different targets downstream of it that help with this attachment process. And then further on, it is also found, aside from kinetochores and spindle pole, it's also important for thus actual checkpoint as well as APC regulation, so anaphase promoting complex regulation. So it must be phosphorylating the APC as well, and it's important for spindle elongation. So look at just the way just one PLK1 is important for all of these different steps in M phase, aside from, of course, the CDKs and cyclins that we know about very well. And then it's also important for the cleavage furrow formation, and then it's also important for the mid-body PLK1 locus, essentially this region when it eventually cuts off. And the other ones as well, they seem to be okay. They seem to be more, localized to one part of the cycle. Like for instance, your PLK4 is mainly associated with centriole duplication, whereas your PLK2, it's more important during the G1S transition, making sure that centriole duplication is taking place properly. As in it is ready for centriole duplication. So I just thought I'd show you guys something other than cyclins and CDKs, and that is one of the examples of that is this polo-like kinase. Right. Another topic I thought I would cover. Oh, does anyone have any questions about PLKs? Of course, you don't I don't think you need to like memorize any of this. It's just it's like an aside, and going into a little detail into one of these things that was not mentioned in that much detail. So, yeah, cool. Anyhow, so if you want to read more about PLKs, you can read about this paper. I think they've also mentioned how because they're so ubiquitous across the cell cycle, it's actually a great target, right? If you have a cancer cell that has some mutation, and PLK1 is very important for the cell cycle to progress. If you could target PLK1, maybe you can figure out a way in which you could halt the cancer cell's progression. So people are always on the lookout, especially when it comes to cell cycle regulating things, these tumor suppressor or oncogenic proteins and genes. People are always on the lookout for a good target or a good therapeutic target that you can focus on when you are working on cancer therapeutics, and trying to make cells proliferate less, make cells sort of freeze in there until they die or something. So I think this paper also includes that. And there are other papers about how PLK1 is, people are looking into how it could be a target in humans, obviously. Yeah. So cool. If no one has any questions, I'll move to the next topic in just a little bit of I won't go into too much detail here, but there was mention of, how the chromosomes, how they have different shapes, right? So based on where the centromere is. So the very common one that you guys see in a lot of your textbooks and diagrams, and also in all of these different images and videos, more often than not, you see this metacentric chromosome. So what does that mean, right? Why is it that the why is actually very interesting, but what are the different ways in which the chromosome can be divided. So typically when people are doing all this karyotyping and all, when they're looking at a bunch of different chromosomes and determining their characteristics, they divide the chromosome into two parts. And even the gene, like the annotations and all, that is done where you have like some specific locus on the P arm and a specific locus on the Q arm. So it's good to know that. So you can divide the chromosome into the P and Q arm, and the point where they constrict, that is the centromere. Okay, and it can be located in different ways. So you can have your metacentric chromosome where your centromere is in the middle, and your P and Q arms are similar in their length. So when it comes to humans, these are some examples. Your chromosome 1, 3, 16, 19, 20. Apparently these are all in the human, they are metacentric. Then you have submetacentric, where the centromere, as you can see, is a little bit off the center. So your P arm is shorter than the Q arm to a degree. And chromosomes 2, 17, 8, 4, all the way to 12, it seems, are all submetacentric. Then you have acrocentric, and this is where it's like much more offset from the center. So P is really, really a lot shorter than Q, and these chromosomes are that way. And telocentric, interestingly, is not found in humans at all. So none of our chromosomes are telocentric, is where actually the centromere is found right at the end of the chromosome. So there's no P arm. So you'd think that it is basically where the telomere is. So it was something that I was thinking about, and I never actually questioned this myself, when I was studying it in, school and college and all, was that why is it that we have these different organizations? What is the point? Why can't all our chromosomes be metacentric? Why is it that there are different um, organizations? Does anyone know? And do any of you guys have an idea why this is the case? We can discuss about it. Anyone have any idea? You can just wildly guess. It's totally cool. Like, it is an open question here. Anyone? You can message if you don't want to talk on the mic. So I don't know really if there is like an exact right answer to this question. To be honest, I think I need to do more reading myself. And maybe you guys, if you're interested, could look it up as well and see really what is the evolutionary requirement for this kind of a thing. Why do we have differently shaped chromosomes? But something I saw as a preliminary kind of look into it was that um, metacentric chromosomes, because the P and Q arm, they are sort of similar in length, pulling these two apart is it's more stable. So essentially there's less chance that you might have a little bit of the chromosome on one side excessively breaking off or some mixing or something. So there's less chance of that. And so similarly, submetacentric also would be relatively more stable in comparison to acrocentric and telocentric. And so why telocentric might be there is just to help with rapid evolution. So it turns out apparently that a lot of rodents, like this mouse, rat, and all, they have a lot of telocentric chromosomes. So that's interesting, right? We don't have it, but mice and rats and other rodents, they have a lot of telocentric chromosomes organized that way, right? So could it be that at some point in evolution, this was what favored their selection? I don't know. I'm not really sure. It's something that I thought it was an interesting question to think about from a sort of a evolutionary biologist perspective. So maybe you guys can think about it, and how about you let me know next class so that I also know more about it. If you guys are interested, you could look it up and find out, and we can talk about it next class. Yeah. So, okay. So, moving on from there. Okay, another topic, right? So, you guys learned in a lot of detail about centrosomes and how they're important for organizing and uh, attaching to the microtubules, right? So I'm not going to go into too much detail because you already know all of this, but essentially, in brief, the centrosomes are composed of these two centrioles, right? And they are arranged perpendicular to one another, and they're surrounded by this PCM, which is the pericentriolar material. Now, no one here has any membrane, so this is all based on, the viscosity and the phase separation of these proteins that allows it to stay there. And the PCM, of course, has a very important bunch of proteins, and it's very important for microtubule nucleation and anchoring, of course. And it has this gamma tubulin. It has also pericentrin and other proteins as a part of it. It's a very, very, very protein dense area. And in general, each centriole of the centrosome is based on this triplet microtubule assembly, right? So there's this cartwheel like structure, and it can contain these different proteins and stuff, centrin, synexin, whatever. And in many cell types, the centrosome actually can be the starting point of a cilium. So you know what a cilium is, right? It's one of those long sort of whip-like structures that can come out from the cell and allow it with help with movement and sensory um, functions, right? So in some types of cells, you can actually have a cilium coming out of it, but as in when the cell starts to divide, the cell will reorganize it and replace it with the centrosome. So it looks like this, right? This cartwheel structure, if you count, there are 9 of these triplets from inside to outside. Each of these little um, subunits, it can be called A, B, and C. Then you have the central rod, which is called the hub, where all of these connecting rods are attached to, or the spokes are attached to. So you have 9 spokes and 9 triplets here, and this entire thing is forming this cartwheel like structure, right? And then you have this repeating, 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 and that is what forms the centrosome. Um, Centrioles of the centrosome, right? And I thought it would be interesting for you guys to see some electron micrographs because that really shows how they look. So I took this from this paper. You can, you're free to go and take a look at the paper yourself as well. So I'll just tell you guys what you're looking at. So if you look at the figure A here, you can see PCM. That means it is the pericentriolar material. So this all these proteins. Now, one thing you should know is that in an electron micrograph, if it is dark, that means it is more electron dense. Okay. So this is an electron dense area. So that means that you have a lot of proteins and a lot of, dense stuff over there that is allowing that causes the micrograph to look this way. So this is a transmission electron microscope image. Okay. Then this is the mother centriole. So as you know, when it comes to the two centrioles, there is a mother one and a daughter one, right? Or a or a procentriole. So this is the mother centriole and this is the procentriole. Okay. And this is like a longitudinal section of them, and they have sort of shown an inset here where you can look at this triplet and how you had seen here. Now that there is an A, B, and C, here also you can see this A, B, and C. So this drawing that they got, it's not from nowhere, it's from real life. This is really how it looks. Okay. um right, and of course, yeah, useful to know is the scale bar. This is 400 nanometers. So this one bar, this much is 400 nanometers, meaning if you look at this entire structure seems to be a little more than 400 nanometers, right? And this guy is maybe like 250 or so. Now at B, what you're seeing is these are centrosomes in, I think, in a cell of a human retinal pigmental epithelial cell. Anyway, that's not important. But essentially, there are two mitotic chromosomes here. Sorry. and they have expanded their pericentriolar material, and here it's labeled, sorry, with a gamma tubulin antibody, and they're positioned on opposite poles of the mitotic spindle, and each centrosome contains these two centrioles, and they've labeled it with this protein marker centrin 1 GFP. So they've used a GFP tagged centrin marker for it. And in this one you have it in interface, so it's not going to be as visible. It's in one end of it. Okay. Oops. Huh. Yeah. And in C, this is basically, I won't go into too much detail, but this is essentially where I had mentioned how it can be the starting point of a cilia, right? So here essentially you have a cilium that is coming out of the cell, and it has these appendages. This is the distal appendage, and this is the subdistal appendage, and this is the cilium in the same kind of cell that was shown over here. So this is just to show that it can be organized in different ways. Okay. And this is actually super resolution imaging that they have done here. It's using a technique called STORM, or stochastic optical reconstruction microscopy. And essentially, it has looked at four centrosomal proteins that have been immunolabeled. So you've used antibodies to label them in different channels or different colors. And they have been then superimposed here. So you can get an idea of the localization of these different protein components. Yeah, with respect to where the centriole is. So they've looked at these different proteins: acetylated tubulin, CEP63, gamma tubulin, and pericentrin. So this is how they localize when you put it all together. Yeah. So I hope that was sort of interesting and sort of putting a face to the name sort of for this structure, because it is very complex. Right. Okay, so we have a few more questions. Sorry. True or false? Centrioles are composed of 9 triplets of microtubules arranged in a circular pattern. So you guys, what do you think? I just talked about this. Is this true?
True.
Yes? Yeah, I think someone said true, though I couldn't hear it too well. So you're absolutely right. It is true. So just as I showed you right now, there are 9 triplets, where the triplets have that A, B, and C notation, and there are 9 of them in a cartwheel structure, right, of microtubules. Right, next question. True or false? Each centrosome in the animal cell consists of a single centriole.
False.
So it actually has a pair, right? So that's absolutely right. This is a false statement. A centrosome has a pair of centrioles, which are then duplicated before cell division. Then centrosomes. Oh, I forgot to write the true or false here. So true or false, centrosomes are always positioned near the nucleus throughout the entire cell cycle.
False.
That's right. So I'll explain why it's false, right? So they actually, initially they are, as you saw in those initial fluorescence micrographs that I showed you, that they were pretty close to the nuclear membrane starting out during interphase and prophase, and then they started dynamically repositioning themselves and moving apart during mitosis, right? Okay, great. Next is, which of the following proteins is essential for microtubule nucleation at the centrosome? So this is part of your coursework, so I think you guys will know this.
Gamma tubulin.
That is correct. Very good. So that is the answer. Gamma tubulin. Yeah. Great. So, all right. The next question is during centriole duplication, the daughter centrioles, right? They begin forming at what angle relative to the mother centriole?
180 degree.
Mm, try again. Does anyone know? So the answer is actually 90 degrees. So I think if you guys remember from your coursework that there was a point where the daughter centriole starts sort of growing out from the foot of the mother centriole, and that was at 90 degrees. So maybe you can sort of revise that once because I don't have a slide to show it actually. Do I? I think here. Actually, we have something here. So this is the procentriole, and this is sort of coming out from here. So you can sort of see it that this is a 90-degree angle over here. Not from maybe this section, but essentially this. If you think of this, since it's a longitudinal section, you can think of this as like one tube, and you can think of this as coming out of the tube below it. So that would make it 90 degrees, right? Yeah, yeah, cool. Right. All right. So then the last question for this slide. All right. So what happens if a cell has extra chromosomes?
Option B.
That is correct. Good job. So that's correct. It may undergo defective chromosome segregation, and that could lead to aneuploidy. So basically, aneuploidy, as you know, is a condition where you have, after division, you can have cells that have an abnormal number of chromosomes. So you can have either like an extra chromosome or a couple of extra chromosomes, or one could be missing. So that if it somehow manages to uh, cross all of the checkpoints and have this extra centrosome and that's not detected somehow, then indeed it can undergo defective chromosome segregation and that could lead to aneuploidy. Of course, I think C option may be. I think that if the cell is at a point where it has extra chromosomes, as extra centrosomes, there's definitely something wrong. So I think the cell cycle probably wouldn't stop because whatever has happened leading to extra centrosomes clearly is not able to detect that there's something wrong with the cell. So the checkpoint mechanisms seem to all have gone haywire at that point, and that would lead ultimately to the cell dividing defectively. Yeah. Hmm. Interesting. So, okay. So actually I wanted to show you guys one video, but I can't download it. So I'll just show it online to you. So I hope this is visible. It's just a short video.
Microtubule destabilizing drugs can prevent polymerization or promote depolymerization of microtubules. Examples include natural agents like vinblastine, vincristine, and colchicine, and synthetic drugs like nicotazole. Colchicine, a plant alkaloid, inhibits microtubule assembly and promotes disassembly of preformed microtubules. Therefore, it is used to treat tumors. Disassembly of microtubules by colchicine prevents new blood vessel formation and destroys existing tumor blood vessels. Colchicine acts by binding to the colchicine pockets present at the interface of free alpha and beta tubulin subunits. During mitosis, the tubulin colchicine complex binds to the growing microtubule and prevents the complete polymerization of the spindle assembly. As a result, Chromosomes remain fixed at metaphase plates and cannot segregate into single chromatids, resulting in the formation of tetraploid cells. Microtubule destabilizing drugs can.
Okay, great. So I hope that everyone could hear that. Oh, okay. Yeah, cool. So that was just a little bit about these different drugs. So, all right, so maybe we can go through these questions now about what you saw. So, because these are useful tools that you would use in your research work as well. A lot of people use nocotazole for, okay, I won't say what because that will spoil the answers, but we'll discuss it after the questions. So, yeah. So the question number 21. So which of the following drugs stabilizes microtubule filaments and then prevents their depolymerization, which can lead to mitotic arrest? Do you guys remember? Okay, just a sec. Let me access the chat. Someone is saying T. T is not an option. But okay, do you mean Taxol? Then someone is saying B and then they're saying A. Okay. So, uh, so basically, all right, so the answer is Taxol. Taxol is the one that actually stabilizes the filaments. It binds to the filaments and prevents their depolymerization. And this is something that's used a lot. This Taxol was taken as an inspirational compound, honestly, for paclitaxel and other cancer drugs. So essentially, you give this drug in cells that are rapidly undergoing cell division. So what happens in cancer, right? You have cells that divide a lot uncontrollably. They've lost the ability to realize that they don't need to divide anymore, right? They lose contact inhibition, they lose the all sorts of checkpoint problems. They have p53 mutations, God knows what else. And in this case, you have a lot of microtubule filaments that are polymerizing and depolymerizing, right? So what would happen, right? You have something like Taxol or its derivatives in these cancer drugs that would preferentially bind to cells that are enriched in actively polymerizing and depolymerizing events. So that's how you can catch these cells that are dividing a lot. So if you have, say, some paclitaxel based chemotherapy, then that would wipe out more cells that are actively dividing than good cells. But of course, it leads to a lot of side effects, a lot of toxicities, a lot of good cells are wiped out, and that's why people become so weak after chemo, as of course we all know. So this is one of those drugs, right? Taxol. It's a naturally occurring compound actually, and it stabilizes microtubule filaments. Okay, next question. Number 22. How do vinblastine and vincristine primarily affect the microtubules? So this was there in the video as well. Anyone, any ideas? You can message or you can say it out loud. So someone is saying A. Anyone has any other ideas? Any other competing answers? No? All good? Okay. So yeah, you're absolutely right. The answer is A. They inhibit microtubule polymerization by binding to tubulin dimers. So see the way in which ultimately they are inhibiting microtubules, ultimately they are inhibiting um, say cell cycle progression or whatever, but they're doing it in different ways. So these also naturally occurring compounds, they bind to the dimers, so they don't allow them to incorporate into fresh filaments. So polymerization here is what you're inhibiting. So if you have a ready-made microtubule that's come up and then you add vinblastine or vincristine to the cell, what will happen is that the existing polymer will eventually depolymerize, because that's what happens. You never have a polymer that's just going to just stay static the entire time. You have actively you're polymerizing and depolymerizing at various ends. But here you will not have any more raw material. You will not have any more dimers available for incorporation. So you'll just end up losing out on your polymerized filaments of, or your yeah, filaments of these microtubules. So that's another mechanism to inhibit or affect microtubules. And the last question, number 23. So which of the following drugs is commonly used to induce microtubule depolymerization in experimental studies by binding tubulin and preventing polymerization? Anyone, any idea? So this is very commonly used in a lot of cell biology labs. You know, a lot of labs use it very, very commonly. Have you guys heard of it? Yeah, another person is active in the chat, so they have said that it's B. I hope you guys, other other people are also there. Huh? So you're right. It is B. It is nocodazole. So it is a very commonly used cell biological reagent, and it can be used as an antimitotic reagent, and it can bind to the beta tubulin um, subunits, and it can disrupt the polymerization and depolymerization of microtubules. So it can hamper cells from forming the spindles, and that's why I think your professor also mentioned it in the coursework, that it is used to synchronize cells at metaphase, because essentially many cells in a culture, suppose you want to perform some study on them, say you have created a drug that can affect the cells at a certain phase and you want to take a readout from that experiment. But if all the cells are in different phases, how can you be sure that the readout you're getting is not some non-specific stuff? Right? So the first thing you need to do is find a reagent that can help make sure all of your cells are in the same phase. So how can you do that? So one way to do it is using nocodazole. So that impairs this metaphase spindle formation and make sure that all the cells come, and then you can wash it out and you can go ahead with your experiment, and now all of them are in metaphase. So then you have a clearer readout of your experiment. Yeah. So it can be used for synchronizing cell division and the cell division cycle, and also it can be used, I believe, to halt transport, and mm, I think also interfere with endocytosis, as far as I'm aware. Yeah, cool. So that is about these important drugs that are important for, making cancer drugs or performing various experiments in the lab. Right. So now we learned a lot about these molecular motors, right? So kinesins and dyneins use microtubules as a pathway, whereas myosin uses actin. And particularly important for cell division is uh, the movement of kinesins across microtubule pathways that allow the formation of spindles and the distribution of various cargo across the cell. So I found this really great video by Ron Vale, who of course is pivotal in the discovery of kinesins in the first place and understanding how they work. So maybe we can check it out, and then that will pretty much be it, because this is going to be a little shorter class today. Yeah.
Just like a busy city, there's constant motion inside of your cells. There's new construction, demolition, and most importantly, transporting goods from one place in the cell to another. Cells transport goods along cellular roadways. To transport cargo along these roads, the cells use motor proteins. Kinesin is one of these motor proteins. If you didn't have kinesin and other motor proteins, you simply wouldn't be alive. All the cells in your body depend upon these tiny motor proteins to organize and power themselves, to divide and multiply, and to communicate with other cells. Let's start with how you began, as a fertilized egg. That egg has to divide into many more cells. All of those divisions require kinesin and many other motor proteins. Development, forming tissues and different parts of your body, requires molecular motion and motor proteins. Every cell of your body requires them for survival. What we know now is that kinesin effectively has two legs, and these legs are able to coordinate a walking motion along a track. That track is called a microtubule, and the kinesin undergoes this beautiful choreographed walking action. While the kinesin is walking along this track, at the other end of the kinesin, it gets hooked up to a cargo. These motor proteins move quickly and efficiently. Relative to their size, they move as fast as a car on a freeway. But they're 4 times more efficient than your car in converting chemical energy into motion. We discovered kinesin in 1984. I was 25 years old at the time and a graduate student. I was interested in the transportation system inside of nerve cells. What makes nerve cells so interesting is that they're extraordinarily long cells. For example, the part of your nerve cell that has the nucleus, where the DNA is located in your spinal cord. But it can extend a very long tube all the way, for example, to your foot. All of the building blocks for that nerve cell are made in your spinal cord, and all those building blocks have to be shipped to the very end of that nerve cell a meter away. There had to be some kind of transport system that was moving these building blocks inside the nerve cell, and I wanted to know how that transportation system worked. After we got this transport to work in this test tube, the hunt was on then to find the key molecule that was responsible for that movement. And we eventually found it, and it turned out to be something completely new that no one had ever discovered before. Watching these movements under the microscope, it was fascinating then to figure out, how does this motor actually work? How does something that's a millionth of an inch in size generate that motion? My lab at UCSF spent about 10 to 20 years trying to figure out the answer to that question. We know about kinesin now, we know a lot about how it moves, but there's still so many fundamental questions that we don't know about how all this motility is regulated, how all of these cargoes know how to go to the right places. There are always new questions that one wants to know the answers to.
Okay. So I hope that was sort of educational and explaining the importance of molecular motors. Um, yeah, so I'm kind of keeping the class still here right now today because I'm not doing so well. So does anyone have any other questions? And then we can wrap up for today. Yeah. So just about, kinesins in general. So it's like among the molecular motors, you have, your kinesins, and then you have dyneins and myosins, as I mentioned, that associate with actin. So kinesins use microtubules, sort of like a train track. to transport various cargo, and of course they have to get energy to do this. It's an energy consuming process because you don't have a motor without um, whatever, some sort of fuel, right? So in this case, chemical energy from ATP is used to drive these different conformational changes that you saw, and that is what generates this force that allows it's something that they I think some researchers at Harvard had called it a drunken sailor walk of the molecular motors across microtubule tracks, because they're sort of placing one foot after another in a funny, like, sort of um, funny way that makes them look like drunken sailors. And dyneins actually use, so dyneins, the other motor protein that use microtubules, they actually drive minus n directed transport, retrograde transport. So that essentially means that from the outer, um, so from the outer parts of the cell, so the more cortical regions, they will move inwards um, in a minus end directed way, and they can transport cargo from outside to inside. And they also help with the movement of cilia and flagella. Meanwhile, myosins, as you've heard of, of course, they drive muscle contraction. So you have muscle movements with the help of myosins that move along these actin filaments, and they drive contraction and also some shorter range transportation. Particularly, a lot of this actin cytoskeleton is enriched in the outer regions of the cell known as the cortex. So, the cortical regions of the cell are highly enriched in branched actin. I actually work on actin a lot. So, now that I think of it, maybe next time I can make up for this time and talk a little bit about actin. That might be interesting for you guys if you're up for it. So there's a lot of branched actin present in the outer regions of the cell, the cortical regions, and that contributes to the cells being able to sense their surroundings, respond to various stimuli, mechanical stimuli. They can determine if the substrate they're on is soft or stiff. They can form various, protrusive structures that can help them essentially probe forward or move. You guys have heard of pseudopodial movement. So all of that involves a lot of reorganization of the actin cytoskeleton. And a lot of the force generating that comes along with that is from the myosin molecular motor that moves along actin. So you have muscle myosin and non-muscle myosin. So in this case, I'm talking about non-muscle myosin. Yeah, so these uh, observations that you saw in the video, many of them were done using just simple microscopy, and there are other techniques that can help you look at just very close to the cover slip, like TIRF, total internal reflection microscopy, that can help you visualize. You notice that there was a couple of clips from where kinesin was transporting like a bead, like an actual bead, so as a cargo. So that's super interesting, and that happens really close to the cover slip. So you'd want a technique that can help you visualize what is close to the cover slip, and you don't want any sort of out of focus light coming from any other plane. So that's a technique that you use, which is called TIRF, which only allows around 200 nanometers worth of visualizing above the cover slip. Yeah. So yeah, anyway, I went into a bit of an aside, but basically you have different types of kinesins that are important, and there is an entire family full of them. I think there are some 15 different families in the mammalian system, and you can have based on the positioning of where the motor domain is, you can have N kinesins or M kinesins or C kinesins, and they can basically drive either plus end or minus end directed motility of the kinesin molecule. Um, and M kinesins are actually the ones that depolymerize microtubules. And That kinesin 13 that I had spoken about many slides ago, that is the weird one that can depolymerize it from both ends. But it's yeah, not present in all organisms, of course. Yeah. So anyhow, I hope that this was interesting for you. Oh, I have one more slide. Hold on, I forgot. Right. So, okay, so this is a technique that was mentioned during your coursework, right? FRAP, fluorescence recovery after photo bleaching, and it was a technique used to look at microtubule dynamics. Uh, so basically, they figured out, right, that during M phase, your microtubule dynamics is actually a lot faster. You have a lot more happening than when the cell is in interphase. So interphase, it's resting. Essentially, you still have metabolic activity, but it does not require as much microtubule related remodeling, right? So how did they look at that? How do you look at diffusion, or how do you look at, the molecular dynamics within a cell? Right, you can't see individual molecules so well, especially not without extremely specialized microscopy setups that you can't always have, and you can't always do these techniques live. There's a lot of these single molecule imaging techniques that have come up, um, that can help you really localize where one molecule is or the other molecule is, but doing that live in, say, maybe a dividing cell, that's near impossible. Okay, so this one technique, FRAP, is actually pretty easy to do. I do it all the time, so it's a really fun technique and I really like it. Okay, and we use it to monitor fluorescence recovery within an area that was photo bleached using a high power laser. So you can study diffusion rates, you can study turnover rates of different proteins, either that are within the cell or expressed on the surface of the cell. So if it's within the cell, you can look at diffusion rates. Why? Because say you have bleached a region. So, okay, maybe I'll explain this. So you have a cell here, and this green is essentially a protein that is expressed across the cell. Either you have, say, given it, like you've put some DNA based construct into the cell that is expressing, say, maybe this is green, so you can say it's GFP, just GFP bound to some other protein all across the cell. Now what we do is, so GFP expresses with at 488 nanometers. So that's the excitation wavelength of GFP. Okay, so that's the light you use to excite that fluorophore. So what you do here is you take this 488 nanometer laser or a blue laser, and you make a little what is called a ROI, or a region of interest. This you do in your microscope software only while you're looking at the cell. Okay, and you put 100% power in that laser and you bleach this little area. So now what does that mean? You have hit the fluorophore with such high power light that actually those fluorophores have got damaged essentially. So they're not going to fluoresce anymore. So you have a bunch of dark molecules that have been created over here because of the bleaching. And then you monitor the recovery in that area over time. So again, you're just doing live imaging and you are actively seeing that what is going to happen now is that you have molecules from other regions of the cell that are going to move and diffuse into this area, and free diffusion is always taking place, right? None of the molecules are static. So you have molecules coming in from other regions of the area, and slowly you'll see that it is recovering. Now, obviously, depending on what you're looking at and the relative abundance of these proteins or the speed at which they diffuse, it doesn't have to be that it will recover back to exactly where it was. Or based on the time scale, maybe you're only looking at it for a couple of minutes of recovery. In that time, you don't see it come back all the way up. But what it tells you about diffusion rates, or what it tells you about turnover rates, is that it can indicate whether something is recovering fast or something is turning over, in the sense proteins at the surface that you have bleached, are they being replaced fast, or are they being replaced slow by other proteins coming in from within the cell and being sort of presented on the surface all over again. So you can look at these different parameters when you are using FRAP. And in this case, in the case of your microtubules, right, in that experiment that was mentioned, they looked at, essentially they bleached a region and they looked at the recovery of polymerized microtubules within that region, and they saw that in the case of cells actively undergoing mitosis, you have much quicker recovery, meaning that your dynamics are faster there. So you're able to recover and make up for the bleached or dark molecules in that region in comparison to in an interphase cell, for instance, where the dynamics are slower because the cell simply does not need as much microtubule polymerization and depolymerization going on. Yeah, so I thought this is a really interesting technique, and it helps show you a lot about um, molecular dynamics, and it helped with understanding about microtubules in cell division. Yeah, so now I'm done actually. So yeah, I hope that was interesting for you guys, and let me know, do you guys have any questions? I can help out with answering them. Uh, Anyone? Any questions? Anyone? Or is everyone good? I hope you found it interesting. Do give me feedback as well if there's more you want me to do more of question answers, you want me to cover more different topics. Is there any topic you specifically want me to cover? Let me know. I can prepare accordingly for your next class, next doubt solving session. All good? Okay, then I think we can wrap it up. Thank you all for joining, and I hope you learned something useful. Yeah, that's all for today then.